Good afternoon, students. So today our lecture is all about gene cloning approaches, and as it is clear to you all, like we are discussing this time in seventh semester the topic that is said to be Introduction to Molecular Biotechnology. So slowly, by covering one by one all the lectures, we are coming to know what is the role that is being played by molecular biotechnology in terms of say your agriculture, right? So today we will be discussing gene cloning approaches, and within the gene cloning approaches we are going to discuss only two. We are going to stress only upon the two main types. The very first, that is said to be indirect, and the second one methodology that we will be discussing later on is all about direct methods of gene cloning. So before I actually enter into the methods, I would like to introduce you all with the term that is said to be gene cloning. So gene is what? It is any base sequence that is present on the DNA, it is said to be gene. But when we call for the cloning, that means a particular gene is being inserted into, say that host into which it is being inserted can be your plant, anything. But right now we are discussing this topic in agriculture, so obviously it is going to be plant itself only. So gene number of copies, when we prepare number of copies from a particular gene. So if this is a gene and if we are preparing number of the copies, that is all said to be the cloning. That is clones are what? Clones means when we have many copies of a single plant, and all copies resembles to each other in all the characteristics. That is what clone. That means a similar resemblance. It is similar in terms of phenotype, right? So this is gene cloning. So what is the purpose of gene cloning approaches? Like once we go for the gene cloning approaches, and as the end of the gene cloning approaches, we can multiply one gene in more number, and then we can often look for improving certain characteristics. Say if the plant is susceptible to the disease, then a gene can be taken from another plant that is no more affected by the disease, and this gene can be inserted into, it can be modified, and later on it can be inserted into the plant, so that that particular plant which was earlier susceptible to the diseases, it is now no more susceptible, and it is now resistant to the attack of diseases or whatsoever it is. So whatever feature you want to improve, that feature can be improved by isolating a particular gene, desirable gene, and then making multiple copies of it, and those multiple copies of the gene, they in turn can be inserted into the host, and here host lies plant. So two methods we are going to discuss today. The one is indirect, that means we are transferring a gene. In gene cloning approaches, we are all going to discuss about gene transfer methodologies, right? And transfer is basically carried by two methods. The very first is indirect, and the another one is direct. Indirect name itself is indicating, that means directly, if say this is the gene, then directly it is not being introduced into the plant. If say this is the plant, then gene is not directly being introduced into the plant. It requires a certain mediator in between, and that mediator is picking up the gene, and it later on it is transferring it to the plant. So this is the indirect method, because gene is not directly being transferred. So this is the reason it is said to be indirect. And indirect methodology is basically carried out by a bacterium that is said to be agrobacterium. And because it is carried by the agrobacterium, so within the indirect we are going to discuss agrobacterium mediated gene transfer. That means the transfer of the gene is being done by a bacteria, and that bacteria is said to be agrobacterium. Second is what, where we don't need any mediator. This mediator is not required, and gene can be directly transferred into the plant, right? So in the case where gene is directly being transferred into the plant, there we are again going to discuss the four methods by how a gene can be directly transferred. The very first is ultrasonication, liposome mediated, electroporation, and particle gun bombardment method. So slowly, one and by one, we are going to discuss all these factors in the details, right? So the very first is indirect method of gene cloning approaches. And in the indirect method of gene cloning approaches, we are going to discuss very first, that is Agrobacterium tumefaciens mediated gene transfer. Before actually I let you know how this gene transfer is being successful with the help of agrobacterium, I would like to introduce you all with the concept like what is Agrobacterium tumefaciens. So it is a natural genetic engineering, and when it is to be used for the transfer of say genes, then in that particular case, it is achieved by two ways. Before I go for this point, I would like to make you familiar with tissue. Then what is meant by explant, right? And then what is culturing. Tissue is what? Group of cells that is performing a particular task is said to be tissue. Explant is the part of the plant. Not part, it is the excised part of the plant that is going to give rise to the whole plant when it is provided all the artificial conditions. Culturing means what? When the explant is placed onto the media, say we have already discussed the media used to be Morashi's and Scoop media, that is based on the name of the scientist itself. So what is done, this explant is excised and it is placed onto the culture. So see, if this is the culture, then culture is altogether consisting of the media, and media is consisting of all the nutrients which are actually required for the growth and development of tissue, say explant. Say now this explant is placed above this media, and when it will start growing, this placing the tissue onto the media, this is actually what it is called as, it is said to be culture. Or when we are in process, that it is said to be culturing, right? So this is culture. Culture is what? Culture is, that is artificial media that is providing. But once we have placed our explant onto the media, it is said to be culturing, right? Very first method is what? Very first method is you have explant, and that gene transfer through Agrobacterium tumefaciens is achieved by co-culturing this bacteria. Agrobacterium tumefaciens is a bacteria, right? So it can be, this is the explant. This is the explant, and explant can be co-cultured with Agrobacterium tumefaciens. This is one method. Second, you go for the plant transformation, right? So this is how Agrobacterium tumefaciens is acting as a natural genetic engineer. Natural means because it is available naturally in the nature, and genetic engineering because it is actually playing a vast role in the modification of genetic content. So this is Agrobacterium tumefaciens. Before actually I come to the process, I would like to introduce you all to its certain characteristics, and characteristics are like very first is this agrobacterium, because we said it is a natural genetic engineering, so it is available in the soil. That means if we talk for its availability, then these are born in the soil and so available in the soil. Secondly, these are gram negative bacteria. If we talk for its shape, then its shape is rod like shape, and they have flagella, locomotory organs on its surface. As a result, it is motile kind of bacteria. So it is available in the soil. It is gram negative, rod shaped and motile bacteria because it is having say locomotory organs above its surface. Very important part of it that it is why it is being utilized in the gene transfer is like this bacteria is responsible for coding a plasmid. Plasmid is actually double standard DNA. So it is this bacteria is actually responsible for coding a bacteria. Sorry, it is responsible for coding a plasmid, and this plasmid is basically called as tumor-inducing plasmid. This is called as tumor-inducing plasmid. The reason behind it is having a part that is said to be tDNA, and this tDNA is actually consisting of the gene that give rise to tumor. So this is the reason this plasmid is said to be tumor-inducing plasmid, and it is abbreviated as tumor means T and inducing means I. So it is said to be tumor-inducing plasmid. So this is a vector. Plasmid is often referred as a vector. So what is to be done? Actually, it has the capability like it's out of a hole. There are many reasons which are present within this tumor-inducing plasmid. I won't be going into detail. Only the fact that I will let you know is this tDNA reason is the reason that can be transferred anywhere else. This is the important segment that can be transferred into the plant. Because what we are looking after the transfer of the gene, and since it is the indirect method, that means it requires a mediator, and here mediator is what? Mediator is an bacteria. And what is the very significant feature of the bacteria? That it has a tDNA region that can be transferred into the host plants, right? So this I have told you this reason, but one drawback with this reason, it is responsible for inducing tumor. So if this reason is to be utilized for the gene transfer, we need to ensure like this reason is no more having the genes that are responsible for the formation of tumor. Because if we transferred this reason along with our desired gene, say this is our desired gene and desired gene is inserted along with this tDNA. And now this is said to be recombinant. This is said to be recombinant DNA because it has DNA of the bacteria as well as it has the DNA that is consisting of the desired gene. So two types of DNA are combined together. So this particular DNA is called as recombinant DNA, right? Well, if we don't remove these genes which are responsible for inducing tumor and a disease that is called as a crown gall disease, then what is going to happen along with the desired expression of the along with the desired expression of the features, desirable features, there will also be the expression of tumor. So what is done? It is very first it is disarmed. Disarmed means it is removed from this vector. This region is removed from this vector and then desired sequence is allowed to be inserted here, right? So this is all about recombinant DNA technology that I have already introduced to you all. And this is the concept behind agrobacterium, that why it is utilized for transferring the desired gene into the host plant. So because it has a tDNA region that can be easily transferred to the host plants. So but the one more thing that we need to know, how this bacteria is reaching the plants. Actually, what happens, these bacteria, when any plant is bounded, that it has met an injury, then the bound region releases an chemical that is called as acetostringone. That is called as aceto. This chemical is known as acetostringone, and due to the release of this chemical, these bacteria which are available in the soil, they are itself attracted towards the plant. Due to this chemical, bacteria is attracted towards the plant, Bacteria is attracted towards the plant, and bee is near the rhizosphere, right? So these are the certain features of the tumefacian. So this is the diagrammatic part of Agrobacterium tumefacian. See, where we have many reasons. Very first reason and the important one that I will be discussing is all about tDNA. Right, tDNA, it has an order where it is getting over, it is lying by base sequence. That is present, the one that is present on the left side is said to be left border, and the another that is present on the right side, it is called as a right border, right? So this is the right border, and it is having three genes: auxin, promoters, auxin, cytokinin, and dopamine. And one among it is responsible for producing the disease that is called as crown gall disease. Then there are number of other reasons, and this is another main reason that is important, a virulence reason, because it is actually responsible for the transfer of this tDNA into the host genome. This is not going to be transferred itself into the host genome. It requires a certain promoter, and here the work of promoter is being performed by the virulence region that in turn transfer this tDNA region into the plant genome. So if we talk for the process, then what is to be done? Very first step is construction of an appropriate gene construct. Like suppose you have certain plant. Suppose you have certain plant with you, and plant is deficient in some desirable feature. That means that particular desirable feature is to be inserted into the plant. So very first step is you go for the construction of the gene that is to be inserted into the T region. After the appropriate gene construct, first step is gene construct. Thereafter, it is to be inserted into the T region, because this is only the region that transfer itself into the host genome. Now you have inserted this is a recombinant DNA now, and agrobacterium is later on cultured with the plant cells, so that plant cells. See, bacha, I have already told you one of the process that is said to be I have told you one step already. I have already told you one step, and in that step, I already told you I have already told you see, transformation I was discussing about. So what is in the transformation? In the transformation, what we are doing, we are keeping a cell, and that cell is further surrounded by the gene that is to be transferred into it. And here, what is that gene? That particular gene is Ti plasmid. And within the Ti plasmid, the main factor is tDNA, right? Main factor is tDNA. Now, what is to be done? So here, the appropriate gene construct. First of all, we have constructed an appropriate gene that is about to give the desirable features, right? Thereafter, this proper particular gene construct, it is inserted into the T region. Just a minute. Say this is the gene construct that is giving the desirable features. On the other hand, we have this tDNA. Now, very first step was the construction of this gene construct. Second is its transfer, that means its insertion into the tDNA region. So this is inserted into the tDNA region. But don't forget, from this tDNA region, the region that was actually responsible for the causing the tumor gene, that is removed, right? This is said to be now the DNA that is consisting of two kinds of DNA. This is called as recombinant DNA, where R is small and DNA, the three letters are in the caps. Now, this recombinant DNA Now this recombinant DNA is to be, this is recombinant DNA, and it is to be placed along with the or near to the plant that is actually to be transformed, into which you want this to be inserted. But here it is not easy, like this is the because it is present in the TI plasmid, and you have placed it near the plant. If you have simply prepared this TI plasmid you have and you are placing it in the near the plant, there is not going to be entry of this constructed part into the plant. What you need to do here, you need to actually, what is the correct way for the transformation to take place? You need to culture these plasmids, right? You need to culture this plasmid along with the plant. Culture is what? That means you are placing it into the media, and within that media there is the presence of the plant, and when these plasmids will be attracted by the plant, then it will be easily uptaken. But you need to maintain the conditions that actually allows the transformation to take place. So after the construction of a particular gene and after its insertion into the disarmed TI plasmid, it is to be placed near the agrobacterium, and later on this agrobacterium is to be cultured with the plant cell, so that transformation takes place. Transformation means there is uptake of this recombinant DNA by the host cell, because normally what is there, if say this is the plant cell, it is not normally going to uptake the DNA, right? It is not going to uptake the DNA. So what we are doing, we are culturing it together, and in the transformation, as we all know, like we are going for calcium chloride treatment, ice cold. The one binds the specifically binds the DNA to its surface and also creates the pores, and once there is the creation of the pores into the cell wall, it can easily penetrate into the nucleus of the plant cell, and this is the way how this agrobacterium mediated gene transfer is taking place. But one thing to be noted, one thing that is to be taken care of is here whole of the vector, or say plasmid, it is not being transferred, it is only transferring its T region, only this T region that is having the desired gene within it, only this part is going to be transferred into the host genome, and the rest part will remain outside only, right? And how this part breaks itself from the rest of the part of the vector, it is actually done by the presence of, it is possible by the presence of Vir region, and this Vir region is only responsible for the transfer of this tDNA into the host genome. So I just hope this agrobacteria mediated gene transfer is clear to all. So it is involving the construction of gene, then construction of the gene, then its insertion into the T region. So this is the T region. We constructed the gene desired, that is giving the desired results. Then it is inserted into the TI, and later on this is placed with the TI plasmid, and later on it is cultured with the plant. So once transformation takes place, with the help of the virulence region, this particular tDNA that is having within it the desired gene, it is transferred into the host genome, and the rest of the part it remains outside only. So see, I told you now why when we are culturing how this segment is, how these bacteria could remain near this bacteria, near this host genome, because this is the whenever there will be injury on the any part of the say plant, then there will be the release of a cytosringone. This cytosringone will induce this Vir region, how it is playing an important role in the transfer of this region into the host genome. It becomes active by the release of cytosine by the plant, and once this cytosine is released, it activates the Vir region that is further made up of many genes Vir A, B, C, D, and so on. And when once this gene becomes activated, it transfer this region into it transfer into this region into the host genome, and it becomes expressed. Right? What we can do? How will be able to differentiate, like this has really been transferred into this host genome? For this, what we do, we often insert one selectable marker within this sequence. We often insert selectable marker. We often insert selectable marker, and what we do with that selectable marker, say that marker say this marker is resistant to, kindly listen carefully, say this marker is resistant to antibiotic that is called as ampicillin. To see whether the cell is transformed or cell is not transformed. Transformed means it has uptaken this desired genome or not. Just to visualize that thing, what we do within, while we were constructing this gene, we inserted a selectable marker into it, and the marker is resistant to ampicillin gene. Now what we did, on the other hand, we prepared a media, and this media is often containing ampicillin gene, and it is this media is poured onto the Petri plate. It is poured onto the Petri plate. Now we cultured here our desired DNA construct, gene construct that is having the gene that is resistant to ampicillin. You have already added here antibiotic ampicillin. If transformation has taken place, then these plants must be containing gene construct, and within that gene construct there is for sure going to be the presence of antibiotic ampicillin. If it is not having ampicillin, then all the cells will die because they cannot tolerate the effect of antibiotic which is present within the media. But if transformation has taken place, then host genome is having this gene. Host genome is having this gene, and it will be able to capable of surviving within this media. But on the other case, if no transformation has taken place, there won't be presence of any ampicillin resistant gene within the host genome, and host genome is no more going to grow because it cannot tolerate the effects of ampicillin antibiotic. So this is the way how we can after expression, how we can just confirm, like particular gene is going to be expressed in the say plant or not. See, this is the basic scenario that is taking place during the agrobacterium mediated gene transfer. So this is the bacteria, and bacteria is encoding a vector, and this vector is having tDNA region, and this tDNA region is only the region that can be transferred into the host genome. This here host is plant, right? Now, but this tDNA is often responsible for inducing tumor. So this segment is disarmed, right? It is taken out, and into here only within this tDNA region is inserted the another gene that is our desirable one. Now this, see, this is being separated from the rest of the part of the vector, and it is being transferred into the host genome. How so? Because when this plant is going to be affected, it is going to release a cytostringone, and as a result of release of cytostringone, this virulence region of the plasmid is going to be activated, and this is only the reason that is enabling the transfer of the tDNA into the host genome. This host genome is that is going to enable the transfer of tDNA, that is consisting our desired gene, and within the desired gene is our a resistant gene, right? Selectable marker. So it will be inserted into the nucleus of the genome and it will be expressed here, right? Another example, another this explanation is, see, we have this agrobacterium tumefaciens bacteria. This is its own DNA, but in spite of the presence of its own DNA, it is often synthesizing a double circular form of the DNA that is said to be vector, right? Or plasmid, anything. Plasmid is referred as a vector only. So we have taken out this plasmid, and within the plasmid, the reason that is of interest is tDNA, right? But the again, as I already repeated, this reason is responsible. As this reason is responsible for the, say, causing tumor, so it is to be manipulated, it is to be modified, and into it is to be transferred our desired gene of interest. What we have done here, tumor inducing genes are removed and our desired gene is inserted into this tDNA region, right? Because this is consisting of our gene is having a desired trait, and the two DNA are since being combined, so it has converted into recombinant TI plasmid. It is later on introduced into the plant cell, right? When it will be introduced into the plant cell, it will express itself to see if, and when it will express itself, then from a small X plant a whole plant can be synthesized. This is one more another example of diagrammatic explanation, where this is the bacteria, this is its own chromosome, and this is extra part of the DNA that is said to be plasmid, which is itself is a vector. So it is cultured. See, bound site will release a cytostringone, and due to this, because it is available within the rhizosphere of the plant, so it will be easily attracted by the plant as cytostringone, and when cultured with it, it will transfer its this constructed portion into the host genome. So this is how a bacteria is acting as a mediator, and it is responsible for the transfer of our desired gene into the host genome. So when it will be allowed to grow into the media, it will there will be the growth of the shoot, roots, and once the shoots grows, roots are available, then it is finally transferred into the soil. So this was all about the indirect method of the gene transfer. Now we will be talking for the direct methods, where we don't require any mediator. Directly it is to be transferred, right? So here we will be discussing the four parameters. Very first is ultrasonication, then liposome mediated electroporation, and particle gun bombardment method. Very first is the ultrasonication. Ultrasonication. Here we are producing high sound waves. Here we are here we are producing high sound waves, and you know, in sonication what we are doing, since say this is the cell, and the cell is surrounded by the cell wall, and cell wall does not allow anything to enter inside easily. So what we need to do, we need to create certain pores into the cell wall, so that anything that is present outside the cell, it could enter into the cell. Because here we do not require any mediator. Here no mediator is required. So this is the reason it is under the category direct gene transfer. This ultrasonic wave mediated transformation, this is also called as a sonication, where this cellular, here we will not focus on the cell wall. Here these cellular membranes are broken. Not broken means there won't be presence of any part of the cell membrane. No, cell membrane will be made, within the cell membrane there will be made pores, and through these pores there will be the entry of say our desired gene construct. But these pores are generated by by creating high sound waves, and as a result of high sound waves there is going to be the presence of pores within the membrane, and through these pores there will be the easy and direct penetration of our desired gene construct. So we can say it is non-invasive way. Non-invasive means nothing is visiting here. As in case of the agrobacterium was visiting, that means it was invading plant genome, and later on it was allowing the transfer of only tDNA segment into the host genome. But here it is non-invasive. That means nothing has to visit the host genome. Directly we need to place outside it the desired gene construct, and this desired gene construct will directly get entered into the host genome through the pores that will be created into the cell membrane as a result of sonication, that is as a result of the presence of high sound waves, that is said to be acoustic waves. So acoustic waves are going to create these holes which are said to be cavitation, and this will, with the help of the cavitation, this will change the features of permeability of the membrane, and as a result there will be direct entry of our desired gene of construct. But in some plants this technique has been successful also. If we talk for one of the research center that is available in the Beijing, that is in the some parts of China, it has been used. By using this technology, it had been easy to transfer the gene in plants like wheat, tomato, and sugar beet. So with this, we come to the process of the ultrasonication. So because the very first step is to transfer the gene. That means we need to change the permeability of the gene, permeability of this membrane. So first step is sonication. Then once the permeability of the membrane is changed, then calli are sonicated and they will be transferred here. And side by side, a control experiment can also be conducted to see whether the plasmid grows or not, right? Then third is the liposome mediated gene transfer. Liposomes are what? Liposomes are microscopically small size particles, and because these are liposomes, so they have membrane, double layer membrane, that is said to be lipid bilayer, which are concentric in the nature. That means the water soluble part will be on the inner side and water insoluble part is going to lie as heads on the outer side. These are the heads and these are the tails. Concentric means tails are on the inner part. If this is a circle, concentric means if this is a circle, then heads are arranged on the outer side and the tails are arranged on the inner side, right? So in this way, This is the way. That means this inner part is this entire, it is consisting of aqueous chamber, and outside non-aqueous part is there. These are also called as lipid bags. How liposome is helping in the gene transfer? Very first step is if the liposomes, they undergo a process endocytosis. So whatever is to be transferred into the cell, that is to be inserted into this liposome, right? And due to the process that is endocytosis, that means it can take out anything that is present outside to the inner side. DNA actually enters the protoplast. This DNA enters the protoplast is the cell that is without cell wall. If say this is the cell, endocytosis, if here is the liposome that is having a desired gene, so it will undergo endocytosis. That means this membrane will fold and it will intake this DNA. It will intake this DNA, right? So very first step before and within the endocytosis, DNA enters the protoplast and it gets adheres onto this is the protoplast here. DNA is adhering, it is attaching itself here, and then it is fusing together, and then it is releasing, right? So this through this liposome mediated gene transfer, success has been achieved in terms of gene transfer in the plants like carrot, tobacco, petunia, etc. So this is the liposome mediated. Very first part is you have to transfer the DNA fragment into the liposome, right? So you are mixing both the parts. So liposome has constituted DNA along with within its part. Now these liposomes are actually responsible for the transfer of the gene in the protoplast. So this will adhere itself to the surface of the protoplast. Later on, as a process of endocytosis, that means this membrane will form a round circle around it, and this will release its DNA that it has combined in the very first step. It will be transferred into the host cell, right? And DNA is finally into the nucleus. Here advantages are less toxicity, because the DNA is here now surrounded by the liposome. So no enzyme that is called as DNAs or RNAs, they can act upon the DNA or RNA, because DNA or RNA it is not available. It is not available to the enzyme. So these enzymes cannot act and these enzymes cannot act upon and break the DNA or RNA. And this is the reason, like for the long time, DNA can be stored in the liposome also. It has been applicable in the various cell types, and the results are like it is highly reproducible. Here I will be discussing one example of sunbeam. Within the sunbeam, we will be discussing about one gene that is called as phaseolin gene. Here, actually the scientists who were present at this corporation, Agri Genetics Corporation, that is in the USA, they were actually they were working on a phaseolin gene, which is said to be the major protein of the French bean. And if we talk for the certain features of this phaseolin gene, and then here we talk for the methionine content, then this methionine content is very low in this gene because food value of bean is reduced now. So this phaseolin gene is transferred to the cultured cell of sunflower, and when they will be cultured, they will grow, they will result into the synthesis of callus, that in turn is going to produce a protein, and this protein itself is called as a sunbeam. So from the fasolin gene fasolin gene, we are actually leading to the its expression within the bean proteins that is called as a sunbeam. This is electroporation. So name itself is indicating in electroporation. Again, we are creating the pores as we were creating in the ultrasonication, but in the ultrasonication, we were creating the pores with the help of high sound waves. But here, the waves are being, pores are being created by using high electricity current, by using electric current, right? High volt electric current. So how much high volt electricity can be supplied? 350 volt electricity can be applied to the protoplast suspension, and this will result into the formation of the pores, and through the pores again there will be the direct entry of our desired gene construct. So here what is being done? Electric pulses are given to the cell. Suppose if this is a cell, then electric pulses is given to the cell, and as a result of these electric pulses, when high voltage current will be given to this cell, there will be the creation of the pores, and through these pores, there will be entry of the DNA or desired gene construct into the host genome. Right? As a result, transformation frequency is increased. So once the transformation has taken place, that means there had been the transfer of gene construct, then we can place it onto the media and we can allow the development of microcalli. And this microcalli can later on be allowed to grow onto the media that is having all the nourishment. So slowly, roots will grow, then shoots will grow, and then the whole plant will grow. And later on, the plants, they can be transferred into the soil. So see, this is the plant cell. Outer is the cellular wall. This is the plasma membrane. We have removed the cell wall because protoplast is the one that is no more having the presence of cell wall. So this will be removed, and left behind will be plasma membrane. We can add foreign DNA into it because when we give high electricity waves, high electricity currents, there will be the creation of pores, and foreign DNA can be allowed to enter into it. So see, this is the protoplast is cultured with foreign DNA. Electric pulses are supplied to this protoplast, and there is the creation of say pores, and later on this DNA will get entered into it, and it will insert itself into the host genome nucleus. So this is electroporation. By using the technique of electroporation, several genes have been transferred in protoplast of say varieties like wheat, rice, petunia, sorghum, maize, and tobacco. And transformation frequency can also be achieved. How we can improve the transformation frequency? We can improve it by using linear DNA, right? See, there used to be the two forms of the DNA. One is the linear DNA, and another used to be the circular form of the DNA. So frequency, like more and more DNA, they get entered into the host genome. That frequency in turn can be increased by using this simple DNA, that is called as the linear DNA, instead of going for the use of this circular kind of DNA. And one need to take care of like heat shock treatment, that is to be given, it is to be given only for 5 minutes, and temperature at that particular time is to be maintained is 45 degrees Celsius, and we need to add polyethylene glycol that will help in the fusion of two genomes, and this much kilovolt per centimeter voltage is to be given. So this helps in the successful transfer of our desired gene construct into the host genome. This is the last is particle gun bombardment method. This was the method that was developed by the scientist named as Stanford and his team. And this term denotes, as a term denotes here, with the help of gun, a particle is being transferred into the plant cell. And there are often existing the another names for this methodology. like particle bombardment, particle gun method, ballistic, microprojectile, and so on. This is basically suitable for those plants from which cannot be regenerated onto the media, right? And where the responses are not as successful, where we when we going for the use of agrobacterium. What is all about this process? See, here is what we have: this chamber. This is the chamber that is totally filled of helium gas, and here used to be the presence of a ruptured disc, right? Below this ruptured disc is placed a plastic plate onto which are present the DNA micropellets, and these are actually the desired gene, and below it is placed a stopping screen, and then we have placed target cell. So these DNA coated cells, which are said to be micropellets, they are actually to be transferred into these target cell or tissue. So how actually this system works? We used to provide a gas cylinder, and slowly and slowly we are filling this we are filling this segment with the helium gas. As the pressure of the helium gas it exceeds the normal pressure, it puts a pressure over here, and as a result this rupture disc is broken. But the pressure is coming with a such and such higher speed that it when this pressure falls onto this plastic micro carrier that is having DNA coated micropellets, then normally when the pressure was not exceeding the normal pressure, this whole process was this rupture disc was available at that particular time, and there was no migration of these pellets towards the target cell. But when the pressure exceed the normal pressure, it broken the rupture disc, and when this pressure fallen onto these when this pressure fallen onto this plastic microcarrier, then it put the pressure onto these micro pallets, and these micro pallets broken the screen which was otherwise stopping it to enter into these cells. It could not stop now because the pressure has exceeded to a larger extent, and as a result it enters into the cells. So this is the direct way of allowing the entry of the micro pallets into the cells, right? So here all what I'm going to discuss about the apparatus, that means we have a chamber. So this one just a minute. So this is the chamber that we have, and it is connected to a outlet so that it could help it in creating a vacuum. Then at the top there is a presence of rupture disc. Then we have a plastic microcarrier that contains DNA coated tungsten particle, and here is the presence of stopping screen, right? So this is the setup. So when we come to the process is like helium gas is allowed to flow into this cylinder at a very high velocity. But when the pressure within this exceeds the bursting point of this plastic dish, then it gets ruptured. Actually, it could control this pressure up to a certain range. But when this pressure has exceeded that particular range, it won't be able to control this, it won't be able to like stop this pressure anymore. And this pressure, when with pressure, this gas falls onto the ruptured disc, it gets broken. When it gets ruptured, this helium gas, it propels like this, like it is propelling in this way. This helium gas is propelling, and then it falls onto this micro pellet. Now stopping screen cannot stop this high pressure gas anymore. And when it is allowed to fall onto this micropallet, the DNA coded, when it is allowed to fall onto this microcarrier, the DNA which are studded within this microcarrier, they get separated from this microcarrier, and later on they break this stopping screen also, and they get inserted into the target cell. So this is the process. That means if cells have uptaken the DNA, they get transferred. Whether they are transferred, this in turn can be verified by inserting one selectable marker into the foreign DNA, and later on they are allowed to grow onto the media. When they will be allowed to grow on the media, there will be the generation of shoot, root, and finally it can be transferred to the soil. With the help of this particle gun bombardment method, success has been achieved in many epidermal tissues of species like alien cepa, scuteller tissues of maize, leaf, cell culture of many crops, etc. And even instead of these plants, even we have inserted the DNA into bacterial cells, algae, fungus, etc. So there are certain conditions that are to be considered while we proceed for this particle gun bombardment method. That is a distance, this much is to be maintained, this much gold size particles are to be taken. Carry distance between the microcarrier and target should be this much. What should be the density? This density is to be maintained. Medium must be supplemented with MS media, and must have 4D, 2 at a level of 2 mg per liter. So these all are the conditions that are required if we successfully want to conduct particle gun bombardment method. So this was all about your today's lecture. So if you have any doubt, we can discuss it right now. So thank you.